Balance is then also another sense related to the ear, but it's definitely not the same as hearing. So before, when looking at hearing, we looked at the cochlea here, which is this rounded shape here. But when we have to look at balance, we have to look at the processing in these semicircular canals and the otolith organs sitting in here. And they also work by the use of hair cells, but within these semicircular canals and otolith organs there is also a liquid, meaning that any rotation of the head or any tilt of the head will be detected by these organs because the liquids inside these organs will flow across these hair cells and either bend them in one or another direction. So for instance, if we look at the otolith organs here, you will see that they are responsible for linear accelerations and decelerations. So if you tilt the head up or down, so you will see that these hair cells in these organs, they will bend and give different signals. On the other hand, if you make a rotation, you will in any directions, you will see that it's the semicircular canals where there are also these hair cells. And then inside these semicircular canals, liquids are flowing. So think of it, if you take a cup, for instance, and have some water and you twist the cup, then the surface of the cup will move compared to the water that you have in the cup. And if you have then some hair cells on the inner side of the cup, then they will bend and thereby be able to, depending on which directions they bend, be able to detect whether your head is rotating in one or another direction. So depending on which way these hair cells are bent, they will either depolarize or hyperpolarize the cells. And one of the more well-studied systems here is the vestibular ocular reflex, which is a reflex. So whenever you turn the head, you will see that your eyes usually is going the opposite way to stabilize the image that you see. And the way that this works is through various brainstem nuclei, and also you have both an excitatory pathway over here. So you will get excitatory signals from the organs going through and activating the muscles for the eye, so that they will stabilize the head. When you make a head turn, in this case to the left, you will have to move the eyes slightly to the right, so you will have to activate the muscles on the right side of the eye. On the other hand, in order to do so and not make co-contractions of the eye muscles, so it just stabilizes, you will also have to have an inhibition of the antagonist muscles on the other side. And this is simply done by this reflex here in the brainstem, which is able to do that. Vision, one of the most important senses for humans, occupying large parts of the central nervous systems in humans. I will not go so much into details about how the retinal processing is, but I will just give you the idea that there are these cones and rods in the retina, and these are spread or distributed differently compared to the center of the fovea of central part of the visual field. And you'll then see that rods are more pronounced to the sides, whereas cones are directly in the center of the fovea. If we look at the central pathways for vision, we have the eyes out here. Then we have the optic tract, or the optic nerve, and we have the optic chiasm here. where you will see that inputs from the two eyes cross and meet. So if we have, this is the left and right visual field, you will see that both information from the left eye and the right eye contains information about the right and left visual fields. And you will see that different parts of the retina will process inputs from the different parts of the visual field. So what happens is that in this case, in this eye, you will have that the left part of the retina process the right part of the visual field. This is also happening here in the left eye. And then you will see that the signals go and meet in the optic chiasm here and is then processed so that signals from the right visual field is projected to the left hemisphere through the optic tract and to the lateral geniculate nucleus in the thalamus, and then further to the primary visual cortex. And this way of having preservation of left and right hemisphere field in the contralateral cortices here is actually something that you will also find when we look at slightly differently, but at least if you look at the visual field here and look at the representation in the primary visual cortex, you will see that we have the left and right visual fields separated into right and left hemispheres, so the opposite hemispheres. But each of these locations, so things that are located close to each other, will actually also be represented close to each other in the cortex. And this is what's called retinotopic mapping of the visual field. So the first time this was discovered was in these recordings from monkeys, where lights were presented to a monkey, and then they looked, they stained for active neurons in the primary visual cortex. And what they could see then was that this very nice grid of lights that was presented to the monkey when they fixated their eyes was actually also present in the primary visual cortex, meaning that any light that was close by each other, they were also close by each other in the cortex. So this retinotopic mapping is a way to actually preserve the map of the visual field in the cortex. And you can actually make very nice maps with functional magnetic resonance imaging of this retinotopic organization of the visual cortex. So the idea is that things that are close to each other in the visual field will also be represented close to each other in the cortex. And the central part of the visual field is mapped in the most posterior part of the primary visual cortex, and it's mapped so that the representation is largest in the primary visual cortex for the central part of the visual field. And you'll then see that you have the upper part of the visual field putting down in the lower part here. So this, and this lie close to each other here, and 2, 6, and 10 close to each other here. With respect to the size, you will see that the central part of the visual field is actually represented in a larger part of the primary visual cortex. And that's also the reason why we see much clearer in the central part of the visual field. And we also have much more neurons in the retina in the central part of the visual field. So that also requires a lot more processing to see the very fine details in the center of the visual field compared to things in the periphery. So this is just a sketch of how the organization of the different layers of the primary visual cortex is. In the visual cortex, you also have cells representations that are direction selective, meaning that if we take a look at what's called simple cells up here, you will see that there is this organization with receptive fields. So in the lateral geniculate nucleus, you have these cells which are which fire when there is light on to the representative part of the retina, and they will then not fire when light is off. And what you will see is that you will have these directions in the primary visual cortex. You will have these direction selective cells. So in this case, you have these simple cells which will fire when there is something that's located in the center of this square here. So they will fire here when there is lights on, and then the surroundings will fire a little when the light goes off. But out here, which is not in the receptive field, you will see that they don't fire when the light is on. You will also can have more complex cells with receptive fields that are responsible when, for instance, a stimulus is moving into this field and then going out, but only in a certain direction. So what you'll see here is that when something is moving into this receptive field at this particular direction, you will see that the cell is firing, and also when it's here and when it's here. But if it's in another direction, even moving into this receptive field, it won't fire. So this is an example of a complex cell, and this is an example of a simple cell. A distinction that's also made between types of cells in the primary visual cortex. And if you then take a lump of cortex and look at it, you will have these columns, orientation columns they're called, where you will see that around this center you will have cells located in this almost spiral shape, where some of them are responsible when, or will fire when there is a light in one orientation, and then it will sort of gradually, when a light is oriented in another orientation, you will see that around this center point here, you will see that the different orientations are represented. So you take a lump of cortex up here, you will see that these nice patterns, they can be found in cortex. Of course, this is difficult to do in humans, but these things can be measured in animals. Then, even for the visual system, there is an even higher specialization in, and subdivisions into primary, secondary visual cortices responsible for more and more complex scenery. So, for instance, if we go to V5 up here, it's responsible, so it's located out here, it's responsible for motion perception. So the higher degree of specialization you have, well, the more complex visual scenes the primary or the visual cortex is able to process. But we will go more into details with that when we look at the lecture on visual perception and other types of perception